Mitochondrial cristae are essential structures within the inner mitochondrial membrane, playing a crucial role in cellular energy production. Here's a breakdown of their composition, reactions, function, and types. The mitochondrial cristae are invaginations of the inner mitochondrial membrane that project into the mitochondrial matrix. Their unique structure is maintained by a complex interplay of lipids and proteins. Lipids. The inner mitochondrial membrane, and thus the cristae, is rich in cardiolipin, a unique phospholipid that is almost exclusively found in the inner mitochondrial membrane. Cardiolipin is crucial for maintaining the high curvature of the cristae and for the optimal function of the protein complexes embedded within the membrane. Phosphatidylethanolamine is another significant lipid. Proteins. A number of proteins are vital for shaping and maintaining cristae architecture as well as for their function. F1, F0 ATP synthase. These protein complexes not only produce ATP, but also form dimers that are critical in inducing and stabilizing the curvature of the cristae. They are often found arranged in rows along the cristae ridges. Micos, or the mitochondrial contact site and cristae organizing system complex, is localized at the cristae junctions, the narrow openings connecting the cristae to the inner boundary membrane, and plays a key role in organizing cristae morphology and maintaining connections between the inner and outer mitochondrial membranes. Key subunits include MIC60. OPA1, or optic atrophy protein 1, is a dynamin-related GTPase involved in mitochondrial fusion and cristae remodeling, working in conjunction with the Micos complex. The prohibitin, or PHB complex, is also involved in maintaining cristae ultrastructure and dynamics. Finally, electron transport chain, or ETC complexes 1, 2, 3, and 4, are embedded within the cristae membrane, facilitating the redox reactions of oxidative phosphorylation. The intricate structure of mitochondrial cristae is directly related to their vital functions. Increased surface area, the extensive folding of the inner mitochondrial membrane into cristae significantly increases the surface area available for the embedding of electron transport chain complexes and ATP synthase. This, in turn, maximizes the efficiency of oxidative phosphorylation and ATP production. Establishment of proton gradient. The cristae provide the membrane surface necessary for the active pumping of protons into the intermembrane space, which is critical for generating the electrochemical gradient that powers ATP synthesis. Compartmentalization is really important here. The invaginations actually create a distinct microenvironment within the intermembrane space, which we call the intracrystal space. This setup can, facilitate the efficient movement of protons to ATP synthase. Regulation of mitochondrial dynamics is, fascinating. Cristae morphology is dynamic and can change in response to cellular energy demands and stress conditions. Proteins like OPA1 and the Micos complex help facilitate these changes, influencing mitochondrial fusion, fission, and, honestly, the overall function of the organelle. The role in apoptosis is, pretty crucial. Mitochondria, and specifically elements within the cristae, are involved in the intrinsic pathway of apoptosis, or programmed cell death, through the release of pro-apoptotic factors like cytochrome C.
While traditionally viewed as lamellar or sheet-like structures, cristae actually exhibit diverse morphologies depending on the cell type, metabolic state, and even stress conditions. The main types include lamellar or flattened cristae are the most common type, appearing as flattened parallel folds. They are typical of cells with high energy demands, like liver cells. Tubular cristae are more tube-like invaginations. They are often found in steroid-producing cells, such as those in the adrenal cortex or Leydig cells, and in some neuronal cells. Vesicular cristae appear as small, sac-like structures and are less common, sometimes seen in specific cell types or under certain conditions.
So, the primary reactions occurring on the mitochondrial cristae are those of oxidative phosphorylation, often called OXPHOS, which is actually the main process by which eukaryotic cells generate ATP. This involves two closely linked stages. First up is the electron transport chain, or ETC. High-energy electrons primarily carried by NADH and FADH2, which are produced during glycolysis and the Krebs cycle in the mitochondrial matrix. are transferred through a series of protein complexes, specifically complexes 1, 2, 3, and 4, all embedded in the cristae membrane. As electrons move down the chain, the energy that's released is used to pump protons or hydrogen ions from the mitochondrial matrix into the intermembrane space, which is basically the space between the inner and outer mitochondrial membranes. Molecular oxygen, or O2, serves as the final electron acceptor at complex 4, forming water, H2O. Next, we have chemiosmosis and ATP synthesis. The pumping of protons creates a steep electrochemical gradient across the inner mitochondrial membrane, and this is known as the proton motive force. Protons then flow back into the mitochondrial matrix through the ATP synthase, which is also known as complex 5, and is embedded right in the cristae. The energy from this proton flow actually drives the phosphorylation of ADP to ATP, which is, the cell's main energy currency.